Hello everyone, welcome to the first episode of our series on cardiovascular physiology. Today we will be discussing the anatomy of the cardiovascular system. We will start by giving you a general overview, then go into the details of the heart, the blood vessels, and finally the lymphatics. Enjoy!
My name is Francesco Sidoti.
My name is Tiziano Russo.
And this is Above Average Med Students, where we make you confident in your medical knowledge. So our cardiovascular system is made of just three components: the heart, the blood vessels, and the blood. So the heart, we all know, is the pump that generates the pressure gradient, which is the driving force for blood to flow in our blood vessels. The blood vessels are we can call them the passageways for our blood to reach all of our tissues, while the blood is the transport medium of nutrients for our tissues, and also, of course, gases that they use to produce energy, for example, O2. Also of waste products like CO2, and of hormones and electrolytes. We know the circulatory system is divided in two circulation, a pulmonary circulation and a systemic circulation. Now, they have different functions because the pulmonary circulation is in charge of carrying blood between the heart and the lungs. And note that all of the blood traveling in this circulation reaches the lungs. And this is different from the systemic circulation because it is in charge of making blood flow between the heart and all of our other body systems. But our body systems are connected in parallel, meaning that the blood that is pumped by the heart in the systemic circulation will be distributed evenly, although this can be regulated, to all of our body systems. Remember also that the heart tissues themselves and the lung tissues, other of course than the air sacs, the spaces in the alveoli, are supplied by the systemic circulation because they need nutrients to function properly. Let's now get into the proper anatomy of the heart, and we can start by talking about where it is located in our body. We know it is in the thoracic cavity, but there is this common misconception that the heart is on the left side of our body. Actually, the heart is bang in the middle of our thoracic cavity. However, we know that it is divided by a septum in left and right sides, and the left side is stronger than the right side because it has the left ventricle and the left atrium, and they pump blood in the systemic circulation, so they are thicker and more muscular. And so when we put our hand on our chest, we usually feel the heartbeat on the left side, but this is just because we feel it coming from the left ventricle, which is more powerful. But actually, remember, the heart is in the middle of our thoracic cavity. Also, there is some nomenclature that refers to the heart that you should know, which is very important. The rostral portion of the heart, the more superior part of the heart, is called the base, which is a bit counterintuitive because you think the base of something is at the bottom of it, while the more caudal portion of the heart, the more inferior part of the heart, is actually called the apex of the heart because it has a pointed shape. So I told you the heart is in the middle of our thoracic cavity, but where exactly? So it is in between the lungs. It is actually connected to the connective tissue of the lungs by means of the pericardium, but we'll see this in more detail in a bit. And it is located between the sternum, which is anterior to the heart, and the vertebrae, which are posterior to the heart. And also note that the heart is a bit slanted. It's not precisely located vertically in the middle of the thoracic cavity, but it lies from top right to bottom left. So the apex is more to the left of the sternum, while the base of the heart, which is superior, remember, lies more to the right of the sternum. So now that we have localized the heart in our body, we know where it is, we know what it is connected to, we can start talking about the heart itself, and we'll get into the details of the layers of the heart.
Okay, yes, so now I'll take you through the actual layers of the heart. Bear with me because it might get a bit confusing. So let's start by saying that there's three layers to the heart. Starting from inside to outside, we have the endocardium, endo for inside, myocardium, myo for muscle, and epicardium, epi for outside. So the endocardium is the innermost layer, and it's divided itself in two layers, the inner and outer. The inner layer is made of endothelial cells, which are a type of epithelial cells that line the vascular system. In fact, it lines the chambers of the heart and the valves. While the outer layer is made of connective tissue. And this is continuous with the myocardium, which is the middle layer. You should know that in this outer layer of the endocardium is where we have interspersed the fibers of the electrical conducting system of the heart, which we will cover in a separate video. Then we have the myocardium, which is the middle layer, and it is made of spirally arranged cardiomyocytes. Cardiomyocytes, as you might understand from the name, are cardiac muscle cells. And how are these cardiac muscle cells different from other striated cells, like those of the skeletal muscles? Well, they are connected by these intercalated discs, which play a very important role in the heart, because these discs provide an attachment, an adhesion between adjacent cells made of desmosomes and gap junctions. If you remember your histology, desmosomes have primarily a structural and support function as they connect to the cytoskeleton of the cells, while gap junctions are basically channels that allow for the free passage of ions. So what does this mean? That thanks to the presence of gap junctions, when a cell generates an impulse or receives an impulse from the previous cell, this impulse can travel seamlessly to the next by passing through these junctions as in the form of ions. This allows for the cardiomyocytes to work as a syncytium, which means a collective of cells that work together as a single unit. You should also know that the thickness of the myocardium varies because, of course, in those parts where the heart will need more force, the myocardium will be thicker, and in the parts where it needs less force, the myocardium will be thinner. For example, in the atria are always thinner than the ventricles because they only have to push blood into the ventricles, while the ventricles have to push blood in circulation. Also, the left ventricle, which pushes blood in the entire systemic circulation, is going to be thicker than the right, which only has to push blood into the pulmonary circulation. So the blood has to go reach the lungs and come back. Finally, we have the epicardium, which is the outermost layer. And the epicardium is the visceral layer of the serous pericardium. What does this mean? It means that the heart is covered by this double membrane called the pericardium, which is divided into two layers, the serous and the fibrous, again, from inner to outer. So the serous epicardium is made of epithelial cells, and itself is divided into layers, again, from inner to outer, visceral and parietal. The visceral corresponds to the epicardium. The fibrous layer is the outer layer of the pericardium, and it is made of dense, irregular connective tissue. The role of the fibrous layer is to attach the heart to nearby structures like the lungs. Between these two layers, the serous and fibrous, we have a virtual cavity which contains the pericardial fluid. In fact, the cavity itself is called the pericardial cavity. So what is the role of the pericardium? Well, we can say it has two main functions. The first one is an actual physical protection from shocks. The second one is to reduce the friction that is generated between the heart and the nearby tissues when the heart contracts. And this is thanks to the presence of this fluid in the pericardial cavity. Okay, so now that we have analyzed the layers of the heart, let's talk about what gives the heart its structure. Well, you should know that the heart has a fibrous skeleton that consists of 4 rings surrounding the 4 valves, which we'll see in a minute. And it also lines the fibrous portion of the interventricular septum. What is the interventricular septum? It is a vertical tissue that separates the left and the right side of the heart. Also more on this later. What is the function of this fibrous skeleton of the heart? Well, the functions are threefold. First of all, it provides a rigid structure for the attachment of cardiac muscle fibers. Secondly, it provides structural support to the 4 valves of the heart. Thirdly, it provides electrical isolation between cardiomyocytes of the upper and lower chambers of the heart. We will speak in detail about this mechanism in a future video. For now, just know that the upper portion of the heart has to contract first, and the lower portion of the heart has to contract later to allow for blood to fill the upper portion, then flow into the lower portion, and then go in the rest of the body. And this is allowed thanks by the electrical isolation provided by the fibrous skeleton. Okay, so let's briefly recap the layers of the heart. Going from inside to outside, we have endocardium, myocardium, epicardium. Endocardium is divided itself in 2 layers. We have the inner layer of endothelial cells and the outer layer of connective tissue. Then we have the myocardium, which is made of cardiomyocytes. Remember the intercalated discs with the desmosomes and gap junctions. Then we have the epicardium. What was the epicardium? It was the visceral layer of the serous pericardium. What is the pericardium? It's the double membrane that covers and protects the heart. And what are the layers of the pericardium? We have the serous inner layer and the fibrous outer layer. The serous inner layer made itself of a visceral layer and a parietal layer, with the visceral being inner and corresponding to the epicardium, and the parietal being outer. Okay, now that we have seen the layers of the heart as well as the fibroskeleton, let's take a look at the organization of the chambers of the heart.
Yeah, so the heart is divided into two sides, a right and a left side by the septum, as he already mentioned. But each side is further divided into an upper and a lower chamber, this time by valves, that we'll talk about more in detail later. The upper two chambers are called the atria, while the lower two chambers are called the ventricles. The atria, the upper chambers, receive blood coming from tissues that is brought by veins that empty in the atria, and the atria will transfer this blood to the lower chambers, the ventricles. The ventricles pump blood away from the heart in arteries. Also remember that the right side of the heart only handles oxygen-poor blood, while the left side of the heart only handles oxygen-rich blood. But we'll now take a look at how blood flows through the heart so you can understand this better. Just before we start, don't fall in the common misconception that arteries always bring oxygenated blood and veins always carry deoxygenated blood. This is just not true. Arteries are vessels that carry blood away from the heart, while veins are vessels that carry blood to the heart. And we'll see that the oxygenation of the blood, whether it is oxygen rich or oxygen poor, and it is traveling in a vein or an artery, just depends if we're talking about the pulmonary or the systemic circulation. So let us now take a look at the journey of the blood flowing through the heart and our circulations. And we start from the right atrium. Remember, the right side of the heart only handles oxygen poor blood. In fact, the right atrium receives blood from two veins, the superior and inferior venae cavae, which respectively collect blood from tissues above the level of the heart and below the level of the heart. The right atrium will transfer blood into the right ventricle. The right ventricle will pump this blood into the pulmonary circulation. Remember, we're still talking about oxygen-poor blood, so the blood has to be oxygenated, and this happens in the lungs. So the right ventricle pumps this blood in the lungs via the pulmonary artery. This artery, almost immediately after leaving the heart, will branch into a right and a left branch so that it can reach right and left lungs. In the lungs, the blood will undergo gas exchange. It will lose its excess CO2. It will pick up new, fresh oxygen. It will become, so, oxygen-rich blood, which is collected by the pulmonary veins, which travel from the lungs to the heart, in particular to the left atrium of the heart, which we said, the left side of the heart only handles oxygen-rich blood. In fact, the blood coming through the pulmonary veins is oxygen-rich. It just underwent gas exchange. From the left atrium, blood is transferred to the left ventricle. The left ventricle, the thickest part of the myocardium, pumps this blood into the aorta, which is our major artery, the biggest one we have, which will eventually give rise to all of the branches of our arterial circulatory system, which will supply with nutrients and oxygen all of our tissues. In tissues, cells will take up O2 and they will use it to produce energy and produce CO2 as a waste product. This CO2 is also collected by the blood, and this blood is now oxygen poor and is ready to return to the right atrium with the superior and inferior venae cavae. So as a recap, we have just described blood going from veins to atria to ventricles to arteries. But you might wonder, how is this unidirectionality of flow ensured? Well, thankfully, we have 4 valves in our heart that have this exact job. We have 2 valves between atria and ventricles, and these are called the right and left atrioventricular valves, or AV valves, and 2 between the 2 ventricles and the respective arteries. So on the left side, we have the aortic valve because it is between the left ventricle and the aorta, while on the right side, we have the pulmonary valve between the right ventricle and the pulmonary artery. All of these valves are pressure operated because they open and close in function of pressure gradients. In particular, they open in the presence of a forward gradient and they close in the presence of a backwards gradient. What does this mean? A forward gradient occurs when the pressure behind the valve is higher than the pressure in front of the valve, leading the valve to open. A backwards gradient will occur when the pressure in front of a valve is greater than the pressure behind the valve and the valve will shut close. Let's make a practical example. Imagine the left atrium. The left atrium is filling with blood, while the left ventricle is yet to be filled and is now empty. In this situation, the atrioventricular valve between the two will be open because the pressure in the left atrium is higher since blood is flowing into it, while the pressure in the left ventricle is lower since it is still empty, and this creates a forward gradient, so the valve is open. Once the ventricle is filled with blood and the left atrium has no more blood to pump, the ventricle will start contracting, and the contraction of the muscles of the ventricle will rapidly increase the pressure inside it, up to the point where it becomes much greater actually than that in the atrium. This will create a backwards gradient because the pressure in the ventricle is much higher than the pressure in the atrium. The backwards gradient will shut the valve close, the ventricles finally can empty into the aorta, and the fact that the atrioventricular valve is closed will prevent backflow of blood from the ventricle to the atrium. Also remember that a backwards gradient can force a valve to close, but will never make it swing open in the opposite direction. This phenomenon is what we know as eversion, and it is prevented by different mechanisms depending on the valve we are talking about. And now Tiziano will take you through the valves in particular, and we'll also see these mechanisms I'm talking about.
Okay, so let's try to build up on what Francesco just said. We will begin by looking at the atrioventricular valves. The name should give it away. These valves are found between atria and ventricles. The right atrioventricular valve is also called the tricuspid valve, and that's because it is made of 3 cusps or leaflets. Remember, tri for right. The left atrioventricular valve is also called the bicuspid valve, guess why? And it is also referred to as the mitral valve. You could remember this as mitral ending with L, which is the first letter of left. These valves are attached to the walls of the heart thanks to some tendinous cords, the Latin name is cordae tendinae, which span from the edges of these cusps, which make up the valves, to the papillary muscles. What are papillary muscles? They are nipple-shaped protrusions found on the walls of the ventricles, which contract when the heart is contracting as well. And the role of these papillary muscles is to prevent the mechanism of eversion, as Francesco said before. How does this work? So, when we have a backward gradient, when the pressure in the ventricles is higher than the pressure in the atria, the cusps of the valves will tend to shift up towards the atria. The reason why they are unable to open towards the atria is because these papillary muscles create an opposite force that pull on the cordae tendinae, which pull on the leaflets of the valves. So if we take a snapshot of the forces acting on a valve upon contraction, we will see the backward gradient, which will push up, and the tension generated by the papillary muscles and the cordae tendinae, which will push down. You could imagine this as a hot air balloon, which is anchored to the ground via some ropes. In fact, the hot air that pushes the balloon up is comparable to the backward gradient generated by the higher pressure in the ventricles, and the cords that keep the balloon anchored to the ground corresponds to the cordae tendinae. Other than preventing eversion of the valves, these cordae tendinae also ensure a tight seal between these 3 cusps upon their closure. So just to be clear, this mechanism of papillary muscles and cordae tendinae only applies to atrioventricular valves. What is the other type of valves that we find in the heart? Well, we have the semilunar valves. These valves are made of 3 leaflets, and they prevent the backflow from major vessels into the heart. In fact, one is found between the right ventricle and the pulmonary artery, and one is found between the left ventricle and the aorta. These valves open during ventricular emptying, which is when the ventricles are contracting. Therefore, the pressure in the ventricles is higher than the pressure in the major vessels. And they close during ventricular filling, meaning when the ventricle is relaxing and the pressure in the vessels is greater than the pressure in the ventricles. How is eversion prevented in these valves? Well, as soon as the pressure in the vessels exceeds the pressure in the heart, the valves shut close because of their intrinsic anatomy, meaning that as we have this backward gradient that inflates these valves, the three leaflets bind to one another and create a tight seal that prevents them from opening backwards. Also, for those of you who might be wondering, there are actually no valves between the atria and the major veins. This is because we don't actually need valves to prevent backflow from the atria into the veins for two reasons. Number one, the pressure in the atria never exceeds significantly the pressure in the veins. Therefore, there is very little backflow to be prevented. Secondly, and this only applies to the right atrium, when the atrium contracts, it actually compresses and sort of closes the opening of the vena cava into the atrium, therefore allowing very, very little blood to go back. Okay, so this is everything you guys need to know in regards to the anatomy of the heart to understand what we will say about the physiology of the heart. Francesco will now give us an introduction to the anatomy of the vascular system.
So remember, at the beginning we said we have the three components of the cardiovascular system: the heart, which is the pump, the blood, which is the medium, and the blood vessels, which are the passageways. And now we're going to get into the details about blood vessels, outlining the different types and the different functions they serve. So, you remember we have the two circulations, the pulmonary and the systemic, but actually they're both made of the same types of vessels, and the sequence of vessel types in both circulations is the same. We have, starting from the heart, of course, arteries carrying oxygenated blood, and the arteries branch into progressively smaller vessels, which are still called arteries, but they're just smaller. A small artery will reach an organ. Upon reaching the organ, it will branch into numerous arterioles. Arterioles are those vessels that travel in the organ and will branch, still inside the organ, into capillaries. Capillaries are vessels that allow for exchange of substances between the blood and the tissues across their walls. Capillaries will merge to form venules, and venules will merge to form small veins that will leave an organ. Remember, a small artery reaches an organ, which branches into arterioles. Then venules converge into a small vein that will leave an organ. So the two vessels that reach organs are a small artery on one side and the small vein which exits on the other side. Small veins join to form progressively larger veins, and larger veins will eventually become the superior and inferior vena cava, which will empty into the heart. Note that arterioles, capillaries, and venules are referred to as the microcirculation, and this is because we can only see them through a microscope. Now let's take a look at what are the similarities and the differences between the several vessel types. Let's start from the most important similarity, which is actually that all of these vessel types are lined with an endothelium layer. As Tiziano said before, the endothelium is a type of epithelium that lines all of our vascular and actually all of our cardiovascular system, starting from the heart, but continuing in vessels too. And the main difference between the types of vessels is the composition of their wall, apart of course from the endothelium, which is what actually makes them specialized for the specific function they serve. Let's start by taking a look at arteries. Arteries are specialized to offer a rapid flow of blood from the heart to tissues. And we know this because they have a very large radius and they offer very little resistance to blood flow. So blood is allowed to flow very fast. Arteries, however, also have another very important function, and it is that they act as pressure reservoirs. In fact, arteries have a very characteristic wall. The endothelium of arteries is coated by a layer of elastin fibers, which is coated itself by a layer of smooth muscle, and then an even more external layer of elastic fibers. The outermost layer is actually a layer of collagen. This layer is what gives the strength to the walls of the arteries to withstand the high pressures of the blood coming directly from the heart. But what we want to focus on is the elastic fibers, because the elastic fibers allow the arteries to act as pressure reservoirs thanks to the phenomenon of elastic recoil. So what is elastic recoil? So let's take a large artery. Let's take the aorta. We know the heart is pumping blood into the aorta. And we know the aorta is eventually going to branch into arterioles. Okay? What happens? A lot of blood is coming in from the heart, while the arterioles are offering, at the other end of the aorta, are offering very, very, very much resistance to blood flow. This resistance to blood flow will mean that less volume of blood is gonna leave the aorta through the arterioles than the volume that is coming in from the heart. So overall, we're gonna have an excess of volume that is gonna stay in the aorta. Okay? I know this is quite hard to understand, but it is such an important phenomenon to ensure a continuous flow of blood in capillaries. In fact, elastic recoil allows the large aorta with a very elastic wall to inflate in response to the excess volume inside it, and then when the heart is relaxing and is not pumping any more blood into it, it will deflate thanks to the intrinsic properties of the elastic fibers. And this deflation is actually a secondary driving force for the blood inside it that will drive that excess volume of blood into the arterioles, which will never be out of blood. So blood flow into the arterioles is always continuous. Note that not all large arteries have the same exact wall. In fact, the arteries that are nearest the heart contain relatively more elastin than smooth muscle, and they're called elastic arteries. While as these major arteries branch into medium-sized vessels and approaching the organs, these vessels will contain relatively less elastic fibers and more smooth muscle, and they're termed muscular arteries. The role of these muscular arteries is not anymore that of pure elastic recoil, but this has more to do with the control of arterial pressure, thanks to vasodilation or vasoconstriction of these large vessels in order to increase or decrease resistance to blood flow. The next type of vessel in the sequence are arterioles. We already said something about arterioles, but let's get into the details of their structure and their functions. Arterioles have a thick layer of smooth muscle around their endothelium, and this smooth muscle layer is coated by a layer of collagen fibers. Note that this collagen fiber layer is much thinner than that of larger arteries, since arterioles have to withstand lower pressures. Also, arterioles present a way smaller radius than larger arteries, and this small radius is enough to offer significant resistance to blood flow, as we already mentioned. This resistance to blood flow offered by arterioles makes it so the pressure exerted by the blood inside the arterioles is lower than the pressure exerted by the blood inside larger arteries. This pressure differential results in a drop of myocardial pressure at the level of arterioles. But more importantly, it serves as a driving force for blood from the heart to the organs eventually. Note that the arteriolar radius can be tightly controlled by contracting or relaxing the smooth muscles of their wall, and this serves two functions. First of all, we can regulate this way the amount of blood that different organs receive, because remember, our body systems are all connected in parallel, so we can close and open different passageways to different systems. Then we can regulate arterial blood pressure. Of course, if we're going to constrict the radius, arterial pressure is going to increase because resistance is also going to increase. The next vessel type in the sequence, as we mentioned before, are capillaries, but Tiziano will take you through capillaries because there's quite a lot to talk about.
Okay, so capillaries are the absolute smallest blood vessel in our body. Keep in mind that they are only 1 micrometer thick. Imagine that the thickness of a hair is 100 micrometers, so they are 1/100th of a hair, and that the diameter of a red blood cell is about 7 micrometers. So this means that red blood cells have to squeeze literally in order to pass through capillaries. The main function of capillaries is that of allowing the diffusion of substances between circulation and tissues. And before going into detail as to how and why they are so good at this diffusion of substances, you should know that they can branch either directly from arterioles and then reconnecting into venules, or they can branch from meta-arterioles, which are these vessels that travel in a straight line, forming a direct communication between an arteriole and its venule. Also, you should know that at the beginning of capillaries, there is the presence of sphincters. What does this mean? That if the sphincters are open, blood will flow from the arteriole into the capillary bed and then back into the venule. Instead, if these capillaries are closed, blood will flow directly from the arteriole through this single metarteriole and into the venule. Whether or not these sphincters are open will depend on a couple of factors. First of all, the metabolic demands of our tissues. For example, if a tissue requires more nutrients and requires to get rid of more waste substances, you will have the opening of these sphincters, as well as for temperature regulation, because if you want to keep a certain part of the body warm, then you will direct more blood towards it. And if you don't care much about the part of the body being cold, then you can shunt blood away from there and direct it into organs and parts of the body with higher priority for temperature regulation. But why are capillaries so good at diffusing substances? Well, we can identify three main reasons. The first one is that they are extremely narrow, as we said earlier. The fact that they are even narrower than a red blood cell forces erythrocytes to squeeze through them, and this makes sure that as much surface area as possible of the erythrocyte is in contact with the walls of a capillary. Moreover, there are billions and billions and billions of capillaries in our body, so much so that their overall surface area of all the endothelium of all the capillaries is extremely large, while the capillaries only contain about 5% of our body blood volume at a time. This means that there is a lot of surface area available for the diffusion and exchange of substances for a very limited amount of blood, meaning that the blood can spread very thin over this large surface area. The third reason is that although capillaries are extremely narrow, since we have so many of them, the overall cross-sectional area of all capillaries in our body is enormous. And as you will remember maybe from your physics class, the speed of a flow of a fluid across a tube is inversely proportional to its diameter, granted that the pressure differential before and after is constant. Meaning that as blood flows from the major arteries to the arterioles into the capillaries, blood will actually slow down. And the fact that blood flow slows down allows for more time for each individual blood cell to interact with the tissues and for substance exchange to take place. Now, we said capillaries are specialized for diffusion and exchange of substances, but you should know that there are different types of capillaries according to the different types of substances that need to be exchanged in different parts of the body. For example, the most common type of capillary is the continuous capillary. Continuous capillaries are characterized by the fact that the endothelial cells that form their walls are tightly attached to one another. So much so that they only allow for very small pores being present at the junctions between multiple endothelial cells, and these very tiny pores allow for only the passage of very small substances like ions, glucose, amino acids, while remember lipophilic molecules and gases can just flow through the membrane of the capillaries. But it will not allow for the passage of large substances. Note that the only part of our body where we don't even have these tiny pores is the brain, because we have to ensure the presence of the blood-brain barrier to isolate the brain from the rest of the body. The second type of capillary that we will analyze are fenestrated capillaries. These capillaries, in addition to the pores that are present between attached endothelial cells, also present these fenestrations, which are larger openings that consist of specialized structures that span through the entire thickness of individual endothelial cells. So, other than the passage of small molecules through the pores, we will have the passage of fluids through these fenestrations to allow rapid fluid movement. This is especially important, for example, in the formation of urine and in intestinal absorption. The third and final type of capillary that we will analyze are discontinuous or sinusoidal capillaries. These capillaries are characterized by the fact that the endothelial cells forming the walls are not tightly bound to one another, but they allow for the presence of large gaps, or also called sinusoids. These gaps allow for the passage of large molecules, and they are especially present in the liver and the spleen. In the liver, because they allow, for example, the passage of protein-bound molecules, protein-bound substances like cholesterol, while in the spleen, they allow for the passage of dead red blood cells or bacteria or antigens in general, because that's where filtration of blood takes place. Finally, we should mention that the permeability of capillaries can be modulated. It doesn't always have to be the same. For example, when there is the release of histamine in the blood, this causes a contraction in the capillaries that allows for more substances to pass through. Keep in mind that, as we already stressed multiple times, capillary walls are only composed of endothelial cells. There are no smooth muscle cells. This means that the widening of these pores and gaps in the capillaries are not achieved by smooth muscles contracting, but by the actual remodeling of actin and myosin in endothelial cells. So, now that we have covered extensively the capillaries, let's move to the next portion of blood vessels, which are the venules.
The next vessel type in the sequence are venules. There's not much to say about venules. They just act as collecting vessels that slowly converge into progressively larger veins, and the small veins will then leave the organ. But you should know venules are made of just an endothelium layer and a layer of collagen coating the endothelial layer. And in contrast to arterioles, which have many important functions, they just have little tone, offer little resistance. So they're just there as passageways. After venules, we said we have veins. Veins around the endothelial layer have a layer of smooth muscles, although thinner than that of arteries. And this smooth muscle layer has interspersed in it some elastic fibers, although they're not very relevant, as they are in arteries, for example. Veins are externally coated also by a layer of collagen fibers and equipped with one-way valves to ensure the unidirectionality of flow in veins. Veins have two main functions. First of all, they act as low resistance passageways for blood from tissues to the heart. This is the exact opposite function of large arteries that acted as passageways from the heart to tissues. They offer low resistance because the total cross-sectional area of veins decreases as small veins converge into large veins, thus speeding up the flow of blood. The second main function of veins is that they act as capacitance vessels. This means that they represent a reservoir of blood in a situation of low metabolic demand for blood. In fact, in resting conditions, veins contain more than 60% of our total blood volume, and remember, as Tiziano said, capillaries only contain about 5% of total blood volume. But how are veins so specialized to be capacitance vessels? Well, veins are very stretchable and very distensible due to the fact that they present relatively more collagen fibers than elastic fibers, and so they have almost no elastic recoil. And this means they can easily distend to accommodate for more volume of blood without affecting venous pressure. Remember that arteries recoil when stretched by an excess of blood, driving the blood forward, while veins simply accommodate for the excess of blood by inflating without affecting pressure. Also note that the blood is not stuck in veins. Like when you think of an excess of blood, don't think that there's just a storage of blood in veins. The excess of blood is allowed to be stored in vein by means of increasing the total cross-sectional area of veins when they stretch. This means blood flow in veins will temporarily slow down so that more blood spends more time in veins. So this is basically all you need to know about our cardiovascular system. We just added a small excursus on the lymphatic system because we think this is the right occasion to mention it, and Tiziano will talk you through it.
So what is the lymphatic system? The lymphatic system is a system of one-way vessels that have the main function of reuptaking excess blood from the interstitial space so that it does not accumulate outside of the blood vessels. Bear with me because it sounds more complicated than it actually is. Why do we need a lymphatic system in the first place? So you should know that at the level of capillaries, as we said before, there is the exchange of substances between the circulation and tissues. This means that there will also be some movement of fluid from inside to outside and from outside to inside of the capillaries. The key thing to understand is that more fluid comes out from the arterial end of the capillary, then will come back into circulation at the venule end of the capillary. Meaning that as blood flows from arteriole through capillary to the venule, some fluid will actually end up leaving these vessels. This fluid ends up where? In the lymphatic system. The lymphatic system is organized much as the circulation in different types of vessels, starting from smallest to largest. However, one thing that you should keep in mind is that it is not a loop, a closed loop like blood circulation. Rather, we have vessels that start at the peripheries that are found in every location of our body that will then converge into larger and larger vessels that will eventually empty in blood circulation. Much like blood vessels, they are made of endothelial cells. However, these endothelial cells have the peculiarity of overlapping, generating these sort of flaps that work as one-way valves, allowing only for inflow of fluid and not outflow. How does this work? Basically, as fluid builds up in the interstitial space, the pressure outside of these vessels will be stronger than the pressure inside of them, so these flaps will open, allowing for the flow of fluid into the lymphatics. Then these initial lymphatics will converge into larger vessels, which are characterized by the presence of smooth muscle. The smooth muscle is important because it confers these vessels myogenic tone. What is myogenic tone? Myogenic tone is the ability of smooth muscle to compensate for a stretch in the muscles themselves by contracting more. So basically, as the amount of fluid inside the lymphatic vessels increases and the walls of the vessels stretch, the smooth muscles will contract to compensate for that stretch. This is actually one of the forces that propel lymph, the name that the fluid takes once it enters the lymphatic vessels, from the initial lymphatics towards venous circulation. The other forces being the contraction of large skeletal muscles around these lymphatic vessels, much like it works for veins, and the pulsatile activity of arterioles, which run also close to these lymph vessels. I have told you how lymph is propelled forward in the lymphatic system. However, how does it not flow backwards? Well, the thing is that, much like veins, lymph vessels also present one-way valves, meaning that as soon as lymph has been propelled past a point, it will not be able to go back. So, just to recap where we started from, we said that at the periphery in every tissue we have initial lymphatics, and that these initial lymphatics will converge into larger lymph vessels. Where do these lymph vessels end up? Well, they end up into two different main lymphatic vessels, the right and left lymphatic ducts. The right lymphatic duct pours into the right venous angle. What is the right venous angle? It is the junction when the internal jugular vein and the subclavian vein unite to form the brachiocephalic vein. Guess where the left lymphatic vessel will pour? Into the left venous angle. So same thing, but on the left side. Just of note, the left lymphatic duct is also referred to as the thoracic duct.
So that is all we had to say about the anatomy of the cardiovascular system. We really hope it was helpful. And if you want to learn more about cardiovascular physiology, you should follow us either on YouTube, Spotify, or Instagram. And we'll see you in the next episode.